Hello, everybody. Welcome to Neurobiology. Hope everybody's doing well. Today, we're going to talk about a field of neurobiology called neuroproteomics. And before we start, I'd like to say that this is going to be an introductory type of presentations. This is just the basic understanding to help us through. Thanks to our group mates here, we were able to gather the information needed to perform out this presentation. What is a neuroproteomics? Actually, neuroproteomics combines two fields of study focusing on the proteomics and neurobiology. And proteomics addresses the proteome study, which is focusing on the set of proteins, the proteome, which is the set of proteins of an organ or an organism under some certain conditions. These protein interactions, whether they are with other proteins or other molecules, they determine the protein function. Thus, these protein functions, or the structure of proteins in general, can be alerted due to changes in the environmental factors like the temperature and the ionic strength. Therefore, we can say that the study of proteomics depicts the dynamics of a proteome under all the possible factors that may affect the organ. Neuroproteomics aims to identify proteins and identify their post-translational modifications, determining how the proteome changes over time and how their genetic factors may change, helping us understand how these changes affect the organism as a whole. Eventually, the objective is to predict and examine how the proteome will progress when it is affected under these certain conditions. Another thing to mention here is that this proteome evolution study carried out through drug designing and molecular manipulation.
After understanding what is a neuroproteomics, let's talk about the neuroproteomics or expression of neuroproteomics. First of all, we should know that expression proteomics, which is also known as protein profiling, is the earliest version of proteomics. What is the purpose of it? The main purpose of expression proteomics is identifying the proteins expressed in a particular sample or tissue under a specified set of conditions and at a particular time. Usually it's compared to expression of proteins in a reference sample. After understanding the aim of this field, let's talk about the most common applied techniques. The first technique is the two-dimensional electrophoresis, which separates the complex proteins mixtures first by charge, as we can see in the figure, and secondly by the molecular weight or by size. So before going to the liquid chromatotandem mass spectrometry, let's mention a note here. These technologies have defined an atlas of the human CSF, cerebrospinal fluid, proteome, which provides an essential source of biomarkers for clinical studies and diagnosis. Liquid chromatotandem mass spectrometry, which combines one or more chromatographic steps with two rounds of mass spectrometry to identify with a high confidence the proteins in a complex mixture. As you can see here in the figure, first we have a protein complex. Typically, a denatured protein complex is digested with trypsin. The fraction of the acidified peptide mixture is loaded onto the SCX column. A discrete fraction of peptide is displaced from the SCX column to the RP column. The fraction is eluted from the RP column into the mass spectrometer, as you can see in the figure. This iterative process is repeated. Obtaining the program's quest is used to correlate the tandem mass spectra of fragmented peptides to amino acid sequences using a nucleotide databases. The filtered output, finally, from the program are used to identify the proteins in a complex. The quantitative methods. The most common method that today we'll talk about in the quantitative methods is the mass spectrometry, which resolves the mass of a molecule by measuring the mass to charge ratio, M over Z ratio, of its ion, which is briefly protein identification. The mechanism of work and the electromagnetic field, as you can see in the figure, we have three mechanism of works. Firstly, the ionization source, then the mass analyzer, and finally the detector. In the ionization source, molecules get ionized by adding or removing charge from neutral species, which makes them susceptible to the electromagnetic field. There are two types of ionizing sources, as you can see: ESI, which is the electrospray ionization, and the MALDI, which is the matrix-assisted laser desorption over ionization. Both of them result, the molecules in both of them get ionized. These ions will be separated either by mass analyzer, either by space or by time. If they are resulted from the ESI, they will be separated by space using quadruple or ion trap. If they are resulted from the MALDI, they will be separated in time by using time of flight analyzer. Finally, we have the detector, which transforms the kinetic energy of the incident ions into a signal current, either by electromultiplier or by photomultiplier with conversion diode. Of course, there are many other applied methods such as liquid chromatography or protein arrays, but the most used one is mass spectrometry.
Moving on with functional neuroproteomics. As we can understand from the title, it deals with the functional properties of individual proteins. And to be more precise, it addresses the post-translational modifications of these proteins and how they are organized into substructures, forming out networks and complexes. New methods have been arising through the study of molecular organization of synapses and neurons as specialized as specialized multi-protein machines and networks of physiological processes can be studied under this field. It also can be targeted for diseases. As the field has been developing lately, new emerging fields in this study has coming out, like the proteomic analysis of protein interactions, which is interactomics. Another field, which is phosphoproteomics, deals with the analysis of changes in the phosphorylation stage of proteins. Synapsoproteomics addresses the study of synapse in the proteomic field. Actually, synapse studies help with neuroproteomic strategies. So researchers focuses on something called the synaptosome, which refers to the isolated synaptic terminal from the neuron, through a process called homogenization, which basically uh, reduces the size of the studied particle to facilitate its distributional study. After isolation of the synaptosome, it's highly recommended to do the protein phosphorylation events to preserve the modifications in the synaptosome due to the highly regulation of signal transmission. As we can see here in the figure, the substructural uh, image of a synaptosome. The synaptosome actually contains the full presynaptic terminal with its all components, like the, as you can see here in the figure, like the mitochondrion and some synaptic vesicles. In addition to the postsynaptical density with its membrane, with some synaptic adhesion molecules between these two. And as a fact here, the first observed and studied synaptosome was in a mouse brain. As a researcher or a scientist carrying out a neuroproteomics experiment, it's highly recommended to follow out these four main steps to get the guaranteed result or some understanding to help us assess the proteomics in neurobiology. So as a starter, the sample must be prepared, and preparing the sample could be from a synapse or a cell structure or a tissue from the brain, processing this tissue to get the protein mixture. Next step is to separate these protein mixture to reduce its complexity. Separation of Separation of proteins could be done through different technologies and methods. Like my friend have talked earlier, we can use the two-dimensional electrophoresis, then we can image our analysis and follow out with next step, which is mass spectrometry. Mass spectrometry measure the data we've got from the separation process, and we can analyze this through different processes. Then we can assess all these measurements and data through computational methods, through protein identification and database search for peptide identification. As we've talked about expression neuroproteomics and functional neuroproteomics, let's talk about the clinical aspects in neuroproteomics. Actually, clinical neuroproteomics addresses the molecular basis of neural diseases, and the neuroproteomics field has been really helpful with discovering unsuspected biochemical changes in the disease samples. Moreover, the evolving technologies has led to new pathways for biomarkers development that helps with diagnosis. And as we have to mention here biomarkers, it involves the identification of a change in a protein that can be used as a marker for the disease or for the disease progression. And some of the fields studied here are brain tumors and psychiatric disorders. As neuroproteomics have been making significant advancement in the neurodegenerative diseases through the study of proteomic methods by the brain tissue, especially Alzheimer's disease. So as we can see in the figure, um, we've talked about this earlier actually, the separation method, whether it's laser microdissection or manual dissection, then following up with the separation and isolation of proteins, then the mass spectrometer to measure these separated data and assessing it finally with the database research of computational methods. Neurodegenerative diseases in neurodegenerative diseases, actually, scientists were able to identify oxidized proteins through neuroproteomics. And in Alzheimer's disease especially, they were able to identify the alerted phosphorylation sites through phosphoproteomics. And to be more particular here, these sites are the microtubule-associated protein tau.
Applications of neuroproteomics. Cerebrospinal fluid (CSF) was the first nervous system sample to have its proteome in 1980s. Currently, neuroproteomics developed to help to identify the biomarkers that can be used to detect diseases. So, let's talk about some newly applied studies or the latest applications. Drug addiction across the synapse, traumatic brain injury. Also, we have the study of proteins' effect on nerve growth, as we mentioned here in a figure. You can see, firstly, the proteins that localize in the neurite tips, and we have the proteins that change the neurite length. And we have the proteins that change the number of neurites per cell. And we also have the proteins that block the neurite autogrowth. Finally, we have the proteins that have no effect on the neurite autogrowth, even though they occur there. Now we can mention the resources and the references. Here we have a list of references that we collected the informations from.
As we have come to an end to our presentation, I would like to thank you all for listening, and I personally hope that this was helpful and beneficial to all of us. Thank you, my friend Malik, for this performance, and thank you, Professor, for this opportunity.
Thank you, Nizar, for this presentation, and I want to thank the listeners, also our professor, for this great opportunity. Have a great day.